PSSM is the data structure that allows us to actually to search with family information, entire family information, in opposing to one sequence. And PSSM is actually derived from the multiple sequence alignment. And in the PSSM, we have frequencies of each amino acid in each position, plus some additional information like gap frequencies and things like that, things like this. And the most popular program which actually makes use of the PSSM is PsyBlast, which we started to talk about. And in PsyBlast, I just remind you, we're actually making several iterations, where the first iteration is like a regular blast search. We come with a query sequence, we search against the database, we retrieve hits from the database in certain threshold of significance. But then comes the difference. Then in opposing to just give the results and say goodbye, we are now continue the procedure. Okay, we take the hit of the first iteration, we align them, so we have now a multiple sequence alignment. Okay, from this alignment we can derive frequencies in each position for all amino acids, and we get the PSSM. And with the PSSM, we can now search again against the database. Okay, but now we have an entire information of the family represented in our data structure, and not only a single sequence. And this turned to be more sensitive, and we get more hits in the second actually iteration, and then we can actually continue in an iterative fashion and make additional rounds, where each time we refine the PSSM based on the results of the last round, and continue to search again against the database, and hopefully we get more hits, and we cover more larger part of the space, of the sequence space of our family. Okay, so this is the motivation. And schematically, I like to do it like this. Basically, when we do a blast search, okay, let's say this is our query, and we did a blast search, so we get several hits in the vicinity of our query, where it's actually fall under some significance threshold that we determined. Okay, let's say we do want more hits from the family. Okay, this is the idea, the entire idea, to get more hits. So theoretically, what we could do is just, let's say, take each of these hits and do another blast search with it, right? Then we could we can advance something like this, okay? Let's say we caught this one. Now using this one as a query, we might catch this one, okay? And so, and hopefully we get more and more hits, and then we can continue. But in PsyBlast, the idea is different. And then it's like we want to catch things in larger circles around the initial point, and not in small circles which go to some arbitrary directions. Okay, and based on all these hits, we can now create a data structure that represents the entire region. Okay, and then with the information which reflect the entire region, we can actually expand the entire space which we might now detect in the next iteration. And now our information is even wider and cover more larger part of the space. And in the next round, we might even catch more hits and things, something like this. Like when we draw, we put a stone in the lake, and something like this. And this turned out indeed to be more sensitive, and it's very good approach. PsyBlast is very popular, and usually when we do a search, we are going to do PsyBlast search because it also includes blast, regular blast, but also give us more non-trivial actually results. And technically, it's actually you can make PsyBlast in the same website that do blast, just to put another V in one of the options or something like this. Okay. So it's really cheap. And over here. Okay, there is one example here.
How does it construct a new matrix? Just based on what it sees, it constructs a new matrix?
Yes.
And, like, it starts with...
It's not exactly a matrix, okay? It's a PSSM, it's not a matrix. Okay? So how he does it, you ask?
Because a matrix is supposed to be something that's true for all.
So this is not a universal matrix, okay? This is in PSSM is a data structure which reflects this family, or a given family. It's not a universal matrix anymore. So we start the search with a regular BLAST search, which uses the universal matrix, BLAST on 62 in the typical case. And then in a second iteration, we are using actually already specific information for our problem, and with frequency which were derived from our family members, which were detected. So this is already oriented to our specific task, which makes it more specific and just better. So here there is an example which demonstrates one particular application or one particular example of success for PsyBlast. And the example is for tRNA synthesizers. These are the enzymes which connect the nucleotides to the tRNAs, okay, before they are bringing them to the ribosome for synthesis. Not nucleotides, and amino acids to the tRNAs, sure. And there is an enzyme which do the connection. And for each amino acid, there is a tRNA synthetase of its own. And the idea here is that, let's say we want to find the relation between tryptophanil tRNA synthetase and tyrosinil tRNA synthetase, which are obviously, they have very similar function, right? They both connect tRNAs to amino acids. Obviously, they have some common ancestral, and they share some of their evolution. And theoretically, it would be nice to find actually this relation by sequence identity. Okay? Problem is that there is only 13% sequence identity between the two proteins. Okay? So a regular BLAST search will not allow us to detect this similarity. And okay, this is example that we know, right? So this is like, we know the answer. But in reality, we'll come with something which we don't know what are the relatives in the universe for this protein, and we want to be able to detect the remote relatives. But here we know that they are related. How do we know that they are related? First of all, they have similar function, and we know that. But also, when we look on the classification of protein structures, you remember I reminded several times that there is a website called SCOPE for classification of structures. Although I haven't shown you that explicitly, but I think I mentioned it. Anyway, in SCOPE, for example, the domains of both enzymes are classified to the same place. It means that there is a very strong structural similarity, which we know in this case. And also, as I said, we know the function. So these two proteins are similar, probably have the same origin, and we want. It would be very nice to detect this similarity in sequence search alone. Okay. But the BLAST of the two protein, or one of the protein, let's say we took the tyrosine RNA synthase, never actually was able to discover this similarity. So this is the top list of the hits which were detected. You see, this is the e-value. Remember that small e-value is better, more significant. Anyway, there are here also hits with non-significant e-values already, so we have here the complete list of good hits. All of them are actually tyrosyl tRNA synthetases. We don't see anything which related to other amino acids. Okay? Actually, there is one here, glutamyl tRNA synthetase. In the borderline, you know, e-value not so good. You see additional. I don't see. But anyway, not tryptophanil. There is no tryptophanil tRNA synthetase in the list. But what we can do now is actually take the top hits, okay? In this case, it will be only tyrosyl tRNA synthetases, but from different organisms. All the tyrosyl tRNA synthetases that turn out to be significant in the first round, which obviously belong to different organisms. So they are orthologs, right? You know the terms already. And we create from these guys, we create a new multiple sequence alignment, PSSM, Okay, then we can run psi-blast iteration, additional plus. By the way, not always we have actually to do it explicitly. We can do all the entire thing in advance if we want, but we can also make any additional round manually if we want. And then we actually run the first iteration. And after, I think, 2 or 3 iterations, all of a sudden the tropophenyl tRNA synthetases start to appear here in the list. See? So we were able to detect with e-value of 10 to the power of -7, which is quite good, that there is significant similarity to between tyrosyl tRNA synthetase and tropophenyl tRNA synthetase. Okay, something that with regular blasts there was no way to achieve. So psi-blast is very nice and allow us to get more sensitive hits for similarity. Yes.
Aren't you entering the bias into your algorithm? So given enough iteration, in the end you will always find something similar.
No, not necessarily. But you are correct that you might put biases. That's correct. So because the database is biased, and if the database is biased and you are now relying on hits from the database, so you are suspected to get biases. It turned out that on average this is more beneficial than damaging, let's say. Okay, but this is something to be aware of, right? This biasing issue, because we are starting to use database information, we are always going to be trapped in what there is in the database. We'll not be able to represent what is not in the database. Okay, still.
After we fished out the tryptophan using the tyrosyl one, shouldn't we be enriching for the tryptophan? Like, are we choosing the tryptophan one and psi-blasting it again and again?
Yeah, but at the minute you detected it, so the question, if you want more members of tryptophan, maybe you can do that. Yes.
If I want a better resolution to the sequence space between the tryptophan and the tyrosyl.
You should remember that in the real application, you don't know what you are going to find. So exactly, okay.
Then after I did find something and I want to enrich it, I want to somehow find it and make something specific to that new entity. I'd rather enrich it and untick the tyrosyl one to get more sequence, better blast alignment.
Okay, if you have some motivation to get specific hits for specific family, you might want to enrich your. That's correct, but I think usually you want to be unbiased and just to find hits or relatives and see what they are. So.
Since you are taking 2 sequences in the first place, BLAST is using the local alignment, right?
Right.
Is it worthwhile using global alignment, so other matrices for global alignments to see?
Local alignment is more appropriate usually than global alignment for several reasons. First of all, proteins may be composed of different domains, and sometimes you want to be able to find similarity in one specific domain. Okay, and the domains could vary, so it's like a combinatorial nature of putting different domains and create a new proteins. But you want to be able to say that even if you have only one shared domain, that these are similar proteins. Okay, and this by definition a local problem already. Although there is also a problem with this domain, I'm going to show it right here. Okay, it's just technical issue. psi-blast is available from several BLAST websites, in particular the NCBI website. The query is just like BLAST-P or BLAST protein. There is one parameter that you may need to specify in addition, which is what's the cutoff of e-value to determine which hits from the first round going to be used in the next rounds. Okay. So this is the only difference in terms of what input we need to provide. And psi-blast we also need to be careful because related to just what we discussed now about biases and errors. So if the hits in the first round are not real hits, this is very bad. Okay, so we want to be sure that we are using correct hits if possible. If non-homologous sequences are included in the PSSM, it's really corrupt the entire search, and the results are not going to be meaningful. Also, we need to remember that if all the hits in the first rounds are very, very similar to the original one, to the original sequence, it's not so good for the search, because we are not using new information to expand our sequence search. This is one problem which related to domains, which I wanted to mention, which is something that we should be aware of. And this is the most maybe a reason why to do global alignment and not local alignment, okay? But still, the reasons to do local alignment, I think, are more important than this drawdown that we have here. But because it's something iterative, what might happen is that, for example, if we have a query with 3 domains, okay, and we detected hits with only 2 of the domains, okay? And then we detected another hits with only 1 domain, so slowly we are getting far and far from the query sequence, okay? And could even be a scenario that the final hits after several rounds are not going to have any shared domain with this query sequence. It's not actually a good example, but could well be, for example, that this, for example, this one we have additional new domain here, and these guys here are only similar to this new domain, so we are not going to find any. It's actually this example exactly. If you consider the ellipse here as a new domain, right? So you see that this hit in the 4th round or something, 4th round, has no shared domain with the query sequence already, but it was detected because of transitivity, because of it has shared domain with the hit in the 4th round, and so this is something which is quite rare, I would say, but it might happen theoretically, and we should be aware of. So it's always good to see in the end also the alignments, where the similarity came from, and to check it, and we do have the alignments if you want to see. So I think we discussed enough. We have also versions to PsyBlast for local use, as usual. So like we can run Blast in local environment, and not on the web. We can also run PsyBlast, which might be useful sometimes to run a lot of searches, or if we don't want to share our sequences in the web. So this was PsyBlast. There are additional tools which make use of PSSM, but this is the most maybe important and popular one. And I think the idea to use, you have to know 2 things. First, about PsyBlast, what it is, how to use it. The second is about this idea of using entire family information for searches and what's funny. Well, this concept of the ellipses that I show you is very important. Okay? No, I don't remember the example. Really, I don't remember it. But this concept is everywhere. I don't know if you see it, but the information in the sequence level when we take family information is more sensitive. Okay, we cover more. We better represent the family. Okay, each family of proteins by definition have the same structure. Okay, and this information in the sequence of the entire family is give us more information about the structure maybe also. And this similarity and the conservation of the structure allow us to model structures if we have another structure of a family member. It's all the time come back if you notice. Now I'm going to talk about several resources which include information about families of protein. So these sites are already, it's not a tool to search like search or sequence search like PsyBlast, but it's already databases, it's already resources that were computed and are out there and we can use them, okay? Information. So it's something that's slightly different, but they are very useful resources. PFAM stands for Protein Family, is the most important database of protein families. Okay, and this database actually include already multiple sequence alignments of all the families available of proteins. The families are represented by HMM, they're constructed by HMM, remind you, hidden Markov model. But you don't, apart of that, you don't have to do any manipulation, or you don't have to use HMM in order to use the data. It's already there, okay? So you can see multiple sequence alignment which were prepared for you. You can download something like PSSM, the frequencies of in each column of the multiple sequence alignment if you want to use them. You can find nice pictures of the logos. We're going to discuss further today how to represent family information with logos. So trees, evolutionary trees, which were derived from multiple alignment, like Ron taught you. So everything which is related to multiple sequence alignment is here for each protein family. Okay, very convenient. There are, you see, about 16,000 protein families as of last year. Technically, they divide the family, the PFAM to PFAM A and PFAM B, which are differ by the quality level of the data. The HMM are generated using a program which is called Hammer. It's a program for multiple sequence alignment using hidden Markov models. And this is what's important. You can also download the models themselves, the parameters, but usually it's of little usage. What's very useful there are the availability of the alignments, okay? Because you can download pre-prepared alignment for your protein. You can come with a new protein to PFAM, see which family it falls in, because you can search there with sequence, for example, or based on the name of the protein even. And then you can download alignments which are prepared for this family. And the alignments you can download either seed alignments, which are manually curated and were the originally sequences which belong to that family, usually several dozens of sequences. And also there are full alignments which include all proteins from all organisms, and these are typically now in level of thousands of sequences. Depends on the task. If you just want for visualization, it might be enough that you have few sequences. If you want to do some statistical analysis on this, better that you will have more sequences. Okay, so it depends on the task. This is a snapshot from one particular protein family in PFAM. So you can see that you can see the sequences, you can see interactions, you can download trees. There are also links to very good programs to show, for example, multiple sequence alignment and to edit the multiple sequence alignments. A lot of formats to download. You can download sequences in FASTA format, in other formats. You see here, for example, in this family, the seed alignment is only 17 sequences, but the full alignment is 2,600. You can define the format of the output, how to see gaps, whatever. So, a lot of options here. You can see trees, okay, which are derived from the alignment with measurement from distances, evolutionary distances. Very nice. So this is PFAM, which give us information about protein families. The length of the multiple sequence alignments, which is there, is on the order of hundreds. Okay, so this is really full domains, sometimes even full proteins. On the other hand, I want to show you another site, which is called ProSite, which make focus on smaller motifs, usually. Okay, so sometimes we are interested in shorter, usually more conserved motif, which have some defined functional role. Someone asked me actually last time, so I think exactly mentioned this one. And the idea here is that we try to see if a given query sequence, contain some known small motifs, which has some significant function. And we can then tell very fast that our protein probably has this function, undergo, I don't know, phosphorylation or glycosylation, because these motifs for these events is very conserved and very clear. Okay, so ProSite is the place which includes such information. The information is not stored as multiple sequence alignment or as PSSM, but in more small structure which are called patterns, okay? Which are, if you're familiar with regular expression, for example, somewhat familiar. So it's like regular expression, it's just short word with letters, sometimes ambiguous letters, which define the motif, okay? Before I show you what a motif is, I want just to show you the interface from for ProSite. So basically, there are two modes for search. You can either come with a sequence. Okay, for example, we have a sequence, we want to know which motifs might be in this sequence. This is probably very frequent use. So we come here to this window and we just copy paste our sequence here and submit, and then we'll get as results all the places on our sequence in which the site was able to detect small conserved motifs. Okay? The opposite is if we know the small motifs and we can search the small motifs and see what's actually the ProSite number of this motif, because each small motif is like a small entry in the database and has its own number. So we can search with this number against the universe of sequences. And we get all sequences in, I don't know, probably in Swissport, because this is European site, which includes our this motif in the sequence, okay? Which might be also useful to see what characterize a given motif, what's the function of the protein which contain the motif and things like this. So it's two opposite searches. One with the motif and find the sequences, and the other we have a sequence and find all the motif.
If you'd like to search by function for phosphorylation, for example.
Yeah, so if we know, for example, that protein kinase A has this motif for phosphorylation, we can come with this motif and see all the putative targets of protein kinase A. Okay? So what is a pattern? A pattern is just a word, okay, which is usually very conserved. So this is a pattern, okay? This pattern includes proline in the first place, valine in the second, alanine in the third, and so on, okay? And the length of this pattern in this case is 6 letters. So this specific pattern identifies one specific sequence, okay? It doesn't have to be like this, okay? Might be that the sequence or our motif is not so conserved. It has some degeneracy, okay? So this is, for example, this is example for something which is more, which is not totally conserved. So this motif, okay, is actually read as follows. This includes argine in all lysine in the first place, okay, because we have this square bracket, so everything within the square brackets is like or, this or this. Then we have X, and X stands for any amino acid. So we don't have any conservation actually in this position. And then we have another amino acid which could be either serine or threonine. Okay? So what's the length of this pattern? 3, only 3. Looks like larger, but this is only 3 actually. And this is very degenerate. So how many possible sequences of length 3 can fall to this thing? We can actually calculate. Here we have 2 possibilities. Here we have 20. Here we have 2. So this is something like 4 times 20, 8, right? And the motif, the pattern could be even much larger than this. You see, this one is probably in the length of, I don't know, 10 or 20. Still not very long, like multiple sequence alignment or something like this, but could be longer than few amino acids. And also there is this regular brackets, and I think regular brackets is like the length. So for example, here there are 3 amino acids, which doesn't matter what's the sequence in this position. And here 4, okay? And in between we have many cytosines. You see it looks like some like zinc finger or something, which needs cysteines in order to coordinate, right? It is a zinc finger. Swear to God, I haven't read it. And indeed, in 3-dimensional space, if we look on this position, so we see that this is the region of the amino acid which fall into this motif in one particular zinc finger protein. Okay? So this motif which we detect on the sequence is usually really reflects something structural also. Sometimes we are not going to know that because we will not have the structure. But usually it should reflect something structural and functional, obviously. How we derive the pattern? So the pattern is derived very similar like how we create actually PSSL from multiple sequence alignment. It's just simpler representation of the information. Okay, so if we have many sequences in alignment, we can go and on each column see what are the letters we actually occupy in this position. And then from this, it's relatively easy to derive already the pattern. Okay? For example, there are positions which are totally conserved, and then it's going to be a single letter in the pattern. If there is some ambiguity, we can put the square brackets. But again, patterns usually are not of entire protein domain, but on very specific functional sites. So the typical length is 10 amino acids, around 10. So this is actually, we showed already, this is actually the old site, I think. Okay, how the results look like when we enter sequence. So we are going to get the post-site ID with some name. For example, this is probably something related to aspergine glucosylation, according to the name. Okay, and if we are going to press on this, we are going to see exactly the typical pattern. What's the definition of the pattern? But in this case, we are in this case we are going to see here where, in which coordinate on our query protein we had this pattern. Okay, so we had two instances actually. In this case, one in position 104 until 107, and one in 144 until 147. And you see that the letters, the two instances are actually different in sequence. This is because the pattern itself is degenerate, so it's not a fixed sequence. Okay. And you see that the same query protein included actually several prosite results, one for glucosylation, one for sulfation, and one for one protein kinase phosphorylation site. So typically we are going to find several structural motifs, several motifs. If structure is available, so we are going to get even links to the structures, and we'll be able to see the the pattern on the structure. But this is only in the instances when we have the structure, which is not, I think, not in the majority of cases. So this was prosite. How we do represent information of protein families, sequence alignment. So we can actually write the alignment itself, which sometimes it's okay. If we have few, not too many sequences, it's all informative actually to see the alignment itself. But in a more general case, we can use sequence logos, which is a very nice graphical way to show information of protein families or of alignment. And you worked in the exercise with this? Yeah. Okay. So you know what it is, right? So it's very nice information, and there is information here about the composition of each column, also about the conservation of each column, because the height of the total of all the letters in the given position, the total height is actually reflect the conservation. And also the nature of the position, because the color indicates the nature of the position. For example, black amino acid represent hydrophobic region. Okay, so all the hydrophobic amino acids are going to have the black color, while, for example, negatively charged amino acids are red, and so and so. So we can learn very fast about conservation, about properties of the alignment, about diversity. So it's very nice, and there are web tools. It's another example of logo. So you worked with a web logo, with some web tool. So you saw that you should come with alignment and get the logo back, right? So very convenient for publications and for research in general. The last resource that I want to mention, I think, in this regard is one which is called Interpro. And Interpro, according to its name, is actually integrated database, which includes all the tools that we just described and more. So we try to gather all the tools which collect information about protein families into one place. So So you'll be able to get results when you come from new sequence to get results based on all these resources. That's why it's actually very useful, because it makes it's a one-stop station for analysis of proteins, of protein families. Okay? So it does include, for example, PFAM and ProCite, but also maybe 10 other resources. And the name of all the resources, I think, is listed in one of the slides, and it's, if not for sure, on their website. And it's a very nice place to actually analyze protein families. Many of the tools are based on hidden Markov models. And I think the list of all resources is given here. Each one of them stands as its own with its own website, but in Interpro you actually can search all of them in one operation. You see here the composition of how many signature or alignments are available from each of the resources in Interpro.